same structures. Okay. But what happens is internalized proteins are broken down into peptides, and then one of those peptide fragments is inserted into a cleft that's in an MHC. This yellow structure here is an MHC molecule. And that is shipped out to the surface of the antigen-presenting cell. So it's expressed on the surface. It becomes an adhesion molecule. And so it has the MHC and that little peptide in that cleft, and that's what the T cell receptor binds. Every T cell, when it's mature, it's able to bind to one type of MHC holding one particular type of peptide antigen. It's very specific. Okay? And it's only, one T cell, one structure that it can bind. And it recognizes both the MHC and the peptide held in that MHC. Now, we'll dwell on that more later. We only have about 35 minutes left, so we need to keep kicking along. Okay, any questions on the surface antibodies and T cell receptors? Because lecture 3 is all on those, okay, MHCs. Those are the topics we'll cover in that short lecture. Okay, going back to lymphoid tissue, so I want to clean up some ideas and make sure you understand about certain lymphoid tissues, in particular the lymph nodes. And I know that you've probably been exposed to this. I'm sorry, that's just a bad thing. You've probably been exposed to this before, but I want to bring up some points. We're going to talk about lymph nodes and the spleen. Okay. First of all, the lymphatic system is connected to the circulatory system. So in time, the fluid that's in the lymphatic system enters the circulatory system. I think right at the vena cava, via the thoracic duct. I'm sorry, right by the heart. So the fluid in the lymphatic system is essentially the same fluid that you're going to have in plasma. Okay, same type of body fluids. The lymphatic system, as you saw on that previous slide, is extensive. The vessels are collapsible. They aren't rigid like capillaries. They collapse. They're more like veins, floppy veins. Okay? And the lymph nodes are present at the branching points of the lymphatic system, primarily along the neck, down the spine, in this area to the armpit, and in the groin area, and down into the thigh just a little ways. Okay? Most of them are along the spine, armpit, and groin. They are locations that, well, this lymphoid tissue is encapsulated, this is connective tissue, and it has lobes. There's connective tissue breaking it out into sections. Each section contains distinctive regions. I'm not going to go through all of those. I don't care that you know about them. But different types of white blood cells are commonly present in different locations in each lobe. The lymphatic system comes in this way, filters through this way, and it exits here. So, dendritic cells that enter via the lymphatic system are going to enter into lobes. And if they have processed antigen, they're going to encounter T cells that are present in that lymphoid tissue. Does that make sense? If there's, not only do lymphocytes bring in antigens, they bring in processed antigens. But if you have an infected cut and it's draining into body fluids, then you may have a lot of bacteria or remnants from dead bacteria filtering into the lymphatics and can enter a lymph node. Does that make sense? And those are the types of structures that B cells may be specific for. Those are free antigens, not processed antigens. And B cells can respond to those, may have surface antibodies for those. B cells and T cells are present in this lymphoid tissue. They hang out there. And they come in through, this way, they hang out here for a few hours and then they exit. they're just trying to go around from one spot to another looking to see where the action is. You know, like high school kids on a Friday night. You know, nothing was happening at the freeze, so you're gonna see what's going on at A&W. I'm sorry to have that correlation, but that's kind of what they're doing. They're going around to see where they can be exposed to antigen and respond. Okay? So, if lymph nodes are the lymphoid tissue that primarily drains the tissues, drains antigen from the tissues. Any antigens that are in the tissues. Usually the nearest lymph node is lymphatic tissue. You know, the skin, connective tissue, muscle, the nearest lymphatic tissue are the lymph nodes. Okay. If an antigen filters in to that lymph node, and let's say some dendritic cells bring in processed antigen to the lymph node. Let's say someone has strep throat. Okay?
Strep throat hurts to swallow, right?
Have you gone to the doctor with strep throat and they do this touchy-feely thing? You know, they look in your throat, they say, ugh. And they do the strep test, and then usually they do this touchy-feely thing. They're looking for enlarged lymph nodes. Because when that antigen, either free or processed, comes in, if there are B cells and T cells that can bind that antigen, they become activated and they start dividing. When they start dividing, they form germinal centers. Germinal centers are like these locations right here. They are dense regions in that lymph node which contain multiple lymphocytes that are dividing. What's that going to do to the size of the lymph node?
Make it swell.
Yeah, it's going to be larger, and you can tell that. People come into the clinic and say, I've got this bump right here. They think it's a tumor. You know, that's instantly what you think. You know, they think it's I have brain cancer. But it's an enlarged lymph node. I mean, you know, you can't, it's outside the skull, you know. But you can feel enlarged lymph nodes. That's a sign of an infection or at least antigen being processed. And bubonic plague. There's a number of infections that cause enlarged lymph nodes in bubonic plague. Those enlarged lymph nodes were called buboes. Buboes. They became necrotic because the pathogen, Yersinia pestis, was there growing and killed the tissue. So lymph nodes are important in infectious disease. Okay, they are a location where lymphocytes are presented antigen that was present in the tissues. Okay, y'all okay with that? Okay, spleen. Spleen is located here, right? Right here. It has two functions. It's actually the lymphoid tissue where antigen that's present in the blood is presented because it is connected to the circulatory system. So it processes antigen from the blood. If you have an IV injection of something, you know, you don't normally do IV vaccinations, but if someone's exposed to something, IV bacterial infection, septic, the spleen's going to be busy. Okay? It's also the location where a lot of old, tired red blood cells are degraded. There's the white pulp, which is the lymphoid tissue, where B cells and T cells are present and where antigen can be presented. There's also the red pulp. That's where old, tired red blood cells are degraded. Okay? So the spleen, can it get enlarged too? You bet. If someone's septic, you know, they palpate the abdomen, you can tell enlarged liver, enlarged spleen. So it's important as a lymphoid tissue also. And someone who's had a ruptured spleen because of some severe trauma, they can have recurring problems with certain types of pathogens like streptococcus. Okay, germinal centers are generated there also if antigen is encountered. Because of the same thing, B cells and T cells are activated and dividing. Now, do they stay there? They stay there for a while, but then they start going into circulation. The lymphocytes will start going into circulation, but they're there for quite a while to establish that clone of activated cells. Okay, this shows T cell activation and B cell activation. This is something we're gonna go through several lectures with this information. And I'm sorry, this is gonna be tedious stuff, but just by way of introduction, here's a T cell. It's being presented antigen by this dendritic cell. The dendritic cell is an antigen presenting cell. Here's the antigen processed and presented on its surface. It doesn't show the MHC complex that holds it. But here's the antigen, here's the T cells, TCR, binds it. That's an important signal. This APC is also secreting cytokines that the T cell responds to. That's what that second arrow refers to. So, you have the binding of the antigen, and then you have an exchange of cytokines. It's kind of like a handshake, and then you're conversing. Okay, and this dendritic cell may actually be secreting a lot of cytokines and actually yelling at that T cell. Okay, and usually there's multiple TCRs binding MHCs with antigen. It's not just one; it's multiple attachments. Okay, and there's other attachments that are occurring too. Oh, we're going to go into those details. Woo-hoo. That's T cell. Okay. I know. I shouldn't get you so excited about this. But we're tempering right now. Okay, if you're into cooking, we're tempering right now. Here's a B cell. B cell, the surface antibody. It's bound antigen. Okay, it may be the same antigen as this little fragment came from. You know, it's red too. You know, so maybe this was engulfed by this dendritic cell and broken down into this little peptide that was then presented to the T cell. Does that make sense? But here we have a B cell. It's bound this antigen with its surface antibody. It's probably internalized some of that antigen, processed it. B cells can be antigen-presenting cells. And it's presented that processed antigen to this T cell, presented a fragment from that peptide, that antigen in its MHC to the T cell. The T cell binds it with its TCR, and then they have this exchange of cytokines. In this case, the T cell kicks out a lot of cytokines to activate that B cell, to stimulate it to divide and to produce bukus of antibody. Okay, so the T cell really does yell and scream at that B cell. Okay, does that make sense? Okay, there's other attachments that occur. We'll talk about that later. These are the three professional antigen-presenting cells. These are the most important ones. These are the most efficient antigen-presenting cells. Other cells can be phagocytes and help clear bacteria, but these are the ones that are very good at engulfing antigen and processing it and presenting it to T cells, to activate T cells. They include the dendritic cells, the macrophages, and the B cells. And I like the series of photomicrograph, electron transmission, and then scanning electron micrograph. Okay, we're going to look at blood smears, so you're going to get a feel for this. Okay, those are the three professional antigen-presenting cells. We'll talk about that more later. Lymphocytes change after they've been activated. They become larger, they have more cytoplasm because they're producing antibodies or cytokines, and so they have a larger amount of rough endoplasmic reticulum, which is the place where proteins that are going to be secreted are produced. The rough is due to the ribosomes attached to the endoplasmic reticulum, and the proteins are released into the lumen of the endoplasmic reticulum, pinched off into vesicles, and eventually make their way circuitously to the plasma membrane where those proteins are released. Okay? Same type of overall process for antibodies that are secreted and cytokines that are one big thing that has to happen is that